We don't believe in, this. I've always said, like, this idea of biohacking. It's a bad way to call it, because it kind of makes it seem like you're doing something you shouldn't be. Like you got your
Yeah.
hand in the cookie jar, like you've gotten to something you shouldn't have ever been allowed to get into. And that's fundamentally the wrong premise. This is your biology. This is your data. You deserve to be in charge of it. You deserve to be knowledgeable of it. Wisdom begins with knowledge of the self. That's Aristotle. And in a lot of ways, that philosophy permeates and tells us.
Paniz, you're one of the big minds in Arizona State University, right? One of the academia guys.
I'm in academia. I don't know if I'm one of the big minds, but yes, Micah. Yep.
For me, probably one of the big minds. So, what is it that you study there?
So I am a postdoctoral research scholar in the School of Molecular Sciences at Arizona State University, and I study mass spectrometry-based metabolomics. Metabolomics is a discipline of systems biology. We look at and study the dynamic relationships of metabolites within various systems. That system can be a human body, it can be an animal, it can be a cell model, it can even be toxicological research from the sewer. That is a system. We look at metabolites across these various systems, and we come up with ideas and mathematical models. And when I say it's mass spectrometry based, mass spec is one of the two technologies mostly used in metabolomics. So you have two camps of people. This is very grossly speaking, those that use nuclear magnetic resonance, or NMR, and those that use mass spec or mass spectrometry for identifying these metabolites. And I happen to use the latter.
So, why'd you land on studying metabolomics?
Yeah, so
I'd say research. Do you say researching, studying? What do you say?
it's kind of the same thing, I guess.
Yeah.
I study metabolomics in the sense that I publish studies on metabolomics. But my research itself is basically for biomedical diagnostics. I got into metabolomics essentially just as a way to study metabolism of lots of diseases. There's the old way where you can, run these kit assays. You get like this very small kit. They come in these little boxes, and you can do, one or even a few metabolites at a time with those kits to study metabolism and figure out, hey, what's going on with a lot of these, a lot of the diseases that we currently suffer from are metabolic syndromes, whether that's cancer, whether that's heart disease, which is the largest killer of humans, greatest threat to human greatest contributor to human mortality is heart disease, right? Alzheimer's, diabetes, these are all metabolic conditions. So I wanted to study metabolic disorder. I wanted to study systems level pathogenesis. And the tools at the time were essentially either, hey, you do these kits, right? And you spend all day doing it, or you can use a mass spec and in 1 hour be able to get hundreds or thousands of metabolites from a single sample.
So, what are the different? You settled on metabolomics as your diagnostic area of research, but what are some of the different forms of diagnostics out there, or how many are there? I don't even know.
We can in systems biology, there's 5 or 6 proper levels, right? Depending on who you ask. It could be the microbiome, it could be the proteome, transcriptome, genome. Even things in the middle now they're starting to do, exposome, whatever the science is conjuring up recently. I chose metabolomics because, okay, the genomics, once we sequence the human genome, that was supposed to be ushering in this new age of precision medicine. And that didn't happen. What we really found out was that the genome is a rough guide for what the body can and cannot transcript and translate. What we really needed to look at was this functional output of the genome. And the lowest level of this that we could do, which is the closest to the phenotype or the disease state or the condition or whatever you're trying to study, that was metabolomics. And there's tons of levels of diagnostics. This is just in systems biology, but like radio imaging, for instance, is a type of diagnostic. Imaging techniques in general are their own set of diagnostics. Blood pressure is a biomarker for hypertension. So like an arm cuff is essentially a diagnostic tool if used to assess hypertension. Diagnostics are kind of all around us, and it's really interesting because they contribute in some ways to this is coming from the Cochrane Review. They contribute in many ways to 70% of all clinical decisions are in some way based on the results of a diagnostic test. But when you look at total medical spending, they only account for 6% of all medical costs. So we have this really great opportunity in diagnostics to invest a little bit and get a lot for that. Our return on investment is huge if we focus on diagnostics.
Yeah, and I mean that makes sense because if you get a good diagnosis, you're going to do the expensive stuff afterwards. For the most part, I know there's expensive ways to do it, but yeah, 70% of you. Sorry, go ahead.
Expensive and invasive. A lot of the things that, for instance, one thing I patented was the first ever complex metabolite test for valley fever, which is a fungal respiratory infection that we get here a lot in Arizona. It's endemic to Southern California, Arizona, and like little swaths in Northern and South America. And we did this test because now when, currently, if you go to take a test, the current, serological or skin antigen testing methods, which are, again, diagnostics, they don't really work all that well. They have about a 70% accuracy, and they fail to work for people that have an immune response. There's a lot of issues of time. So it takes 2 weeks to diagnosis. It can take upwards of $2,000 for just a basic fungal culture. There are instances, horrific instances of people with valley fever who go into the doctor's office and their imaging shows nodules in their chest, very much consistent with lung cancer. So the surgeon will go ahead and do a thoracotomy and open up the thorax of this patient, and they'll realize it's not lung cancer. It is a fungal pathogen. And so we can take all that away with our test, which requires either a blood draw or just a urine collection, totally non-invasive. And it has, between, for urine and blood measurements, it has a 93 and 99% accuracy respectively. And so that is the promise of, sort of these personalized or precision diagnostics, is that we can come up with newer, faster, cheaper, less invasive, more accurate tests that essentially render current standard of care obsolete.
Yeah, and metabolomics is getting down to the raw data. So you said there's like 5 or 6 diagnostic areas in systems biology. Is systems biology, that's just referring to like the internal mechanics of the body, and there's different sets that you would look at, right?
Exactly. So it just refers to the central dogma of biology, which is that you have this DNA that gets transcribed and translated into proteins, right? So we have genomics, transcriptomics, and proteomics. We look at all the genes, all of the transcription, and all of the protein expression profiles that happen. So we have 3 levels there, right? And then we also have the microbiome, which is like this 30-pound commensal organ that, is essentially this great bioreactor and interacting with your nutrition and your environmental exposures. And it is a very important thing to consider. And a lot of interaction effects that we see in health are related around the microbiome. And then very, very down on that, we have the metabolism. So yes, you're right. Systems biology is just compartmentalizing the central dogma of biology from the gene down to the expression of metabolites.
And you feel like would you need some of the other, if you did a metabolomics test and you get to look at however much of the metabolism that's happening, do you still feel like there are times that there's need for other tests like on the microbiome or on the genetic, or does that, because it's so far downstream, kind of cover the bases?
So it depends. And it kind of works vice versa, whereby sometimes, the metabolomics is better than the higher levels of systems biology, and sometimes it's reversed. So, for instance, let's say for metabolomics, we see really high levels of short chain fatty acids in your metabolites. That could be a cause for concern. It could not. It would really depend on whether or not we see a few dominant strains or taxa in your gut microbiome. And so in certain instances, if we see, you have really high H. pylori and you have really high short chain fatty acids, we could then say, hey, you should probably be looking at reducing your composition of H. pylori. We know it has associations with gastric cancer, and we know that those associations are most likely taking place because we have the metabolite output. In some ways, it can add a layer of information that gives us this added confidence and allows us to dial in with astonishing granularity our recommendations or our inferences of what's going on in the body. Sometimes it's not necessary at all that we go to metabolomics. If you have the gene for Huntington's, you will necessarily develop Huntington's and die a very terrible early death because of the penetrance of that gene. That gene has very high penetrance, very deep penetrance. And so there is no need to do a metabolomic screen. Again, giving the, going back to the other hand that of the case, that's not usually the case with the genomics, right? Usually, if you have a gene for something, it asserts a very broad probability of whether or not you will develop that condition. Schizophrenia, for instance, is a great example of, there's a few really well-known genes that are associated strongly with schizophrenia, but it is not, a one-to-one relationship perfectly. And so it really matters, your epigenetic interactions and changes that are then reflected in your proteome and your metabolome. So just taking your genomic screen is not helpful. Or it is helpful, but it's not 100% accurate, I should say. And in the case of breast cancer, if you have the BRCA1 gene, or in the case of Alzheimer's, if you have an APO44 allele, you have a higher chance of developing breast cancer and Alzheimer's, statistically speaking. Whether or not you have the markers is to be determined. And that's not something that genomics can currently answer or ever will answer.
So you've said precision diagnostics a couple times, which insinuates that there is non-precision, not so precise diagnostics. Considering that and what you're researching, and you're obviously super passionate, and I loved our conversations because I really you see a future of medicine where metabolomics is going to radically change how diagnostics are done, as I understand it. I can't speak for you, but what are some of the issues that you're seeing in diagnostics and how they're currently done? I know that's such a big global thing, but just going off of that, what do you think?
No. That's a great question, actually. So the issue currently, and this is bemoaned at length in my doctoral dissertation, was that at least in metabolomics, I can say what the issue is. Globally speaking, I'd also like to say that I envision a world where systems biology in general and this entity that we're trying to form that will curate this body of knowledge, not just for its customers, but for the benefit of humanity. This is what's important. Metabolomics, of course, I'm biased. I do think it's important. I think there's a lot of studies that show when looking at phenotype or output, metabolomics accounts for a lot more variance than the microbiome. It accounts for a lot more variance than the genome. So it is more important, actually, mathematically it is. Is it, however, sufficient on its own? Absolutely not. And ultimately, we would need to scale to all these, different levels of systems biology to get that sort of insight. And I think that if we get that with the correct metadata from our customers, there is no way we can't lead rapid vertical advancements in health and science. The issue with metabolomics, at least, and why we haven't gotten there yet, is manifold. One thing, it goes to regulatory and scientific issues that are very broad and sort of out of the control of many researchers. One thing is a skewed scientific incentive, for instance. Translational work to take the knowledge of metabolomics and translate it to actionable results for humans is very difficult. That pipeline is an arduous many-year-long thing. Now, when you live in a system that says, publish or perish, you have to get out your papers and publish them. There is not enough time, and there's not enough incentive or reward for the researchers that stay behind in the trenches to do that translational work. What happens ultimately is that we get stuck in this endless cycle of discovery. We have this endless cycle of biomarker discovery, but without translation, there is no test at the end. There is no diagnostic. There is no point of care test. There is no in vitro diagnostic. That doesn't exist. There's tons of other issues that abound in the literature as well. For instance, a lot of studies don't report absolute concentrations. We need to get absolute concentrations. We can't get relative response, because what you measure on your instrument is going to give you a different relative response than what I've measured in my instrument. We need to have absolute concentration. So, in blood, that's micromolar, and in urine, it's micromolar per creatinine. We have different, and we all need to get on the same unit essentially, so that I know that if your blood levels of, let's say, tryptophan are dangerously low to a certain concentration and not just a relative abundance from your machine, then you may have some association with depression or anxiety. That is key. There's a lot of things that are holding us back. That's just a couple. The most, I think, the most striking thing is what I asked myself the other day was, the best reason for this business is by asking yourself two questions. First question, is our current ability to measure the levels of human systems, the systems levels of biology, is our ability to measure the metabolome, the genome, the transcriptome, is that greater than the current centrality of all our biological knowledge? And that answer is true. We have a far greater ability to collect and analyze this data than we actually have a well-curated, centralized database of systems biology. That does not exist. There are a few data sets from industries that have 2,000 to 7,000 people, whereby 2 or 3 levels of systems biology has been concomitantly analyzed and collected. But there is no database that currently has, let's say, all 5 levels for 30,000 people. And so that's the first question, that our methods to collect data far exceed our available knowledge. The second question is it easier to assemble that data from scratch than it would be to collect disparate pieces of data from databases or companies and mine that data for insight? Absolutely. It is absolutely easier to do it from scratch than it would be to collect all the pieces and try to piece together some knowledge base. That is the whole reason we started, Therion, is because this has, I think, incredible value to the customer. And also in terms of science, nobody is doing something like this, whereby we commercialize this really great scientific endeavor. You ordered 23andMe, and one of the reasons they asked you why you ordered it was, do you want to contribute to genetic research? I am assuming some people order this test. They spend $100 to $200 of their money because they want to contribute to the research. That is actually a really good, I think, get. It's a really good hook, because you are doing two things. You are getting a ton of insights that are personalized and tailored to your biology, but also that data is being stored and will later be analyzed for coming up with patterns among these systems levels that currently we cannot infer. There is no database like what we are proposing to build. And then if you, like we said, you sit an AI on top of those data, and in a very short matter of time, that AI will be able to pick out patterns. You have this bacteria with this gene and this metabolite, you will have, with a high level of certainty, this outcome. And that is currently not being done even in the science. We have the ability to do it. We have the methods. We don't have the data. And so what we're trying to do is that.
It's kind of because this isn't like new technology, right? You've said this bit has been around since when?
The first metabolomics paper was published by David Wishart in June 1998. And
Okay.
so
So
very new. It is very new. And this, when you read the literature, this is what's being called for. The scientists are saying, hey, why don't we just start a new database?
well,
Why don't
somebody's got to do it, right?
we
Because
try to
they're
do
all
it?
using it as a means to an. I mean, it sounds like more of it's a means to an end. Like you're doing a study and you need a metabolomic test, but you're not like, hey, I want to build the metabolomic framework for people to use, which is kind of the future you're seeing, as I understand.
Exactly, exactly. People are already, so it's a means to a quick end. It's a quick means to a quick end. Writing a paper could be as short as 3 months. Good papers take years, but a lot of people will use this as sort of just a platform to get some data that they can say is significant between their experimental groups, whatever those groups were. What I'm saying is that, hey, there's a ton of these, hey, let's go get them. Let's go analyze this data. Let's go collect samples from, 30,000 people. And let's get all this wealth of biological information. The issue is that like that funding from the NIH isn't there yet. NIH is always like 5 years behind what scientists are thinking and what we want to see happen in the field. And the structure of academia is such that your research, some people excluded, but for most professors is tied to federal funding. The federal government sets the agenda for funding, thereby setting the direction of science. Most science that is done in academia is done through federal funds, right? And so you are writing these grants to calls for projects, funding announcements, and those funding announcements, they set the agenda for the science, and the agenda for the science is still 5 years behind. And also when it gets there, people will say, okay, we want to give a $23 million grant here, a $40 million grant there, and all that grant essentially goes to waste. That grant does not get put into some sort of system or pipeline that is the most efficient way of getting these samples to people and getting results. I think in the business structure where the markets are a better differentiator of your success, that is totally possible. It is a totally self-funded system. It is sustainable. It doesn't need $40 million of government funds. It doesn't need 15 administrative assistants. It doesn't need 5 social psychologists. We need people to work on the social aspects of health. We need to know what kind of data we want to collect from their diets, their physical activities, their depression scores, mental health. These are all data, metadata, we call it, that will be associated with their metabolomics. And if we collect the standardized set of metadata from all of our customers, and when this database starts growing, not just in the number of customers that are in that database, but the levels of systems biology on which we have curated their data, then we can mine it with our machine learning algorithms, our AI general adversarial network that we're building, to find those patterns that would have otherwise taken, like I said, minimum of 3 months for a basic study, many years for many studies. We can do that many times every day. That is the ability, that is the wonder of large data with this incredible modern computing power, is that we will be able to reach incredible insights very, very quickly. And we can, patent that. We can sell those patents to drug companies that are, working on drug targets or whatever, and we can hopefully advance science and medicine, because we're really in a deadlock. If you look at innovation in science and truly paradigm shifting publications, we've decreased like every year, like 9% since 1950. If you look at like where we are, it's been 10 years that people have been really going towards this multi-omic paradigm, and we still are not anywhere near that. So I think the best opportunity to do that is in the business world.
Yeah, I mean, the government, as far as their spending, they're a tenth as effective as the private sector. So, like every $10, or every $10 spent, like the private sector would use that so much better. Yeah, do you see that as like a good setup though? Like, I mean, so they're always like bureaucratic. They're always behind, regardless of the branch. Maybe not military, but there's always some areas that are pretty far behind. Just when it comes to working with the government. Do you see like you're kind of suggesting a different route than probably most of your colleagues or peers would suggest?
Yeah,
Um,
yeah.
do you see a space for this to become more normalized where it's going into the commercial market prior to looking at funding?
Um, to get it into the commercial market, we will probably need some funding. What we have right now is, you order your Aristotle test from Ethereum, you get the test kit, you prick your finger, you put it on your dry blood spot, and then you send it back to our lab for analysis, and then we give you a PDF when your results are done. Ultimately, that is really kind of like inefficient way of doing things on the back end, at least. Doing things at home, taking your blood sample at your kitchen counter is far more simple and easier for people than getting dressed, going down to the doctor's office, sitting in his waiting room, then sitting in his exam room until he or she comes in, and then they give you this whole thing. And it's this very, for me at least, even as a researcher, it's a very awkward interaction. It's time consuming, and it doesn't ever leave me feeling good, to be honest. And that's not to say that I think the idea of medicine is not really good, or that physicians have no place. Of course they do. Of course they do. It's ridiculous to say otherwise. But I think that the issue that we have here is that like medicine and science are at a way where you go to your doctor's office, you go through all of these things, and you get like 19 parameters back. You get a Chem 19 panel, you get 19 parameters back. They might add another 12, give you like, hey, you got 31 panels here. We can give you 126 different blood parameters collected from a dry blood spot at your kitchen counter, mailed back to us so simply. And that I think is attractive too, the convenience of it. And then we give you, a report, hopefully, if we, go this to, right now we're in phase 1 beta testing, but if we take this to the markets, we'll have an app. And that app you can curate and not just learn about what is, your, metabolic profile, but also what you can do about it. Hopefully there will be, we can have develop a way to schedule consults with medical professionals. We're talking physicians, not scientists, physicians or clinical nutritionists, various relevant medical staff, and discuss your results and also learn more about systems biology. I love, I always talk about the app Robinhood that lets you open up and like learn some things about investing and also invest. And that's kind of what I would like, our app to be. We do currently have some of that, extra enrichment materials and learning resources baked into our PDF report. But if you could have something where, you see your levels of glycine are high, and the description of glycine also has a link to glutamate, which you are low in, click that glutamate link and have this sort of interactive platform to connect with your own data and then learn about systems biology on top of that, that would be ideal for me. And it would be a really easy way for us to update our reports, because currently I can't update a PDF. I have to update the whole thing and send it to you. But we could say, hey, as we learn more about systems biology via our own database or from reputable studies that are being published, we can update your profile based upon your most recent results. And that is huge, and that kind of offers scalability with an app.
I think that's an incredible concept.
One thing
Oh man, I loved it.
Yeah, he loved it. He's on, and he's like, at least Jasby's honest. There's a lot of actors in this space, and I will not name them because I don't want this to be, hey, I'm kind of throwing shade at our would-be competitors. These actors in this space are not run by scientists. They are run by VC MBA types in a business suit. They are so far from trying to actually deliver research science to people who are in need of it. It's a gimmick. It's a fun thing. It's it's not scientifically cogent, and we at least are doing something honest, and that's that's kind of where I think Tom got the name, because they all have these like really impersonal names that sound like generic like prescription drug names. You know what I mean? Like they'll have something that'll be like some Simovar and cyanocell, and these weirdly like impersonal names that sound like more like medical or research suppliers than like your health testing company. And so, we're not stuck on the name Ethereum, but he's proposing Jasbi. I think it's wrong because of two issues. First, I think I'm uncomfortable putting
Yeah.
myself front stage in anything, really. I am kind of a reserved person. And the second issue is that, this is so much bigger than me. This is so much bigger than metabolomics. This is so much bigger than, the Human Genome Project. This is the promise of the Human Genome Project. And I would want the name to be ampliative. I would want it to be encompassing not of where we currently are, but where we strive to be one day, and leave room in that name to accommodate our potential growth.
Yeah. I liked Jasby. I mean, I know for you it's probably harder, but the word or the name wasn't even about you. I think the fact that it's your last name, right? So it coincides with you a bit and would be somewhat mentally inseparable. But I think I just liked the sound of it and how easy it is to say, to be honest. Like I didn't really think of it much beyond that. So I know I don't get a vote on it, but if I did,
no, you definitely get a vote. You 100% get a vote. By my count, there's 4 people that get a vote, and you're one of them. And, yeah, no, you 100% do. And you know what's weird is that the weirdest, most amusing outcome usually happens, right? And so this thing will probably be called Jasby in 3 months, and I'll be okay with it. But right now, and for the time being, I like the idea of Prometheus. Prometheus, of course, was the Titan who stole fire from the gods of Mount Olympus, and taking pity on humans, brought them fire. And that fire, of course, is symbolic of technology. And in many ways, that is what we are trying to do. There are lots of people in academia, in medicine. There are tons of people at the maybe NIH or FDA that would not be so enthused about this prospect. Of course, that's where the science is going. Of course, that's what I think the innovators and people who have a much longer time horizon are looking at. But there's the bean counters. There are the paper pushers, and those guys, I think, are hesitant. I think a lot of what we're doing, this sort of global approach, this sort of global reconciliation of the knowledge we have. We want to formalize and consolidate our knowledge to translate results for people. What are we doing in academia and science if not translating results for people? What is the point of the science? And so there's a lot of people that see our global approach as a direct threat, as an attempt to undermine their sort of hey, I work on breast cancer and metabolomics. Hey, I only work on Alzheimer's and metabolomics. And that's great. And we need those people, of course. I'm not advocating against that. But they feel threatened, and I think in many ways. And I think they I don't really tell a whole lot of people about what we're doing or what the goals are, because it's a very big goal. And it is very accomplishable. It's difficult, but it is not impossible. We have not set our eyes on anything that is remotely nearing impossibility. And yeah, so going forward, and there's a lot going on. For instance, there's the Valid Act, which is going through Congress right now, the House and the Senate. And, that act has the potential to really change the landscape of in vitro diagnostics, which is the service that we offer, is an in vitro diagnostic. Even if we don't necessarily say you have this thing, you have that thing, we are a laboratory developed test operating in that sphere. So the landscape is shifting. I think in a lot of ways it's shifting for the better. There's in the Valid Act, there's some language that makes sure that our instruments are within a certain level of precision. That previously was not a regulation, that was not a requirement. I think it's great to get standardized precision between labs as a fundamental barrier to offering IVDs, in vitro diagnostics. But there's a lot of things in that kind of make it really impossible for us to ever seek some sort of like full like insurance reimbursement. There's, HSA we could potentially seek out in the future, but it limits our basically our distribution channels, and it makes it a out-of-pocket test, which I think is well worth it. If we get this tested at a couple $100, it is far less than, about a third of your cost of going to your doctor's office and without insurance and getting 19 parameters in your blood, many of which are not analyzed statistically in the ways that we do. You're not, yeah. Go ahead.
So would we, are there diseases that wouldn't populate on the metabolomics test?
Yes and no. So yeah, there are. Our metabolomics test, our Aristotle test, monitors 126 sentinel metabolites, and they're sentinel because they are strategically chosen to give us wide metabolome coverage. When we talk about metabolome, we're talking about like all of the metabolites in your system. There are more than 25,000 identified, not necessarily, quantified, but detected metabolites in the human blood. And you might think, okay, you're going after 126. That's not a lot. In the context of some research applications, it might not be a lot. In the context of current standard of care, it's over 10 times as much. And in the context of metabolism, it is very appreciable still, because those 126 metabolites, like I said, are strategically chosen to be in various pathways such that we can monitor 68% of all known human metabolic pathways with at least 2 metabolite entries in that pathway. So with 126, and this is really taking advantage of the redundancy in biology. One metabolite could be involved in 5 or 6 pathways. Another metabolite could be involved in the same 5 or 6 pathways and not involved in anything else. But with including those 2 metabolites, we have reliable detection of 5 or 6 pathways. And so you look at all the pathways in the human body that are currently quantified. And you look at how many we have and where they're situated. And like I said, 68% of known human metabolism can be monitored with our panel. Now think about how,
So what's a contrast to that? Like what would be?
The contrast to that would be like 5% with your current doctor's visit.
Yeah, right. So we're getting a much more comprehensive view at it.
Exactly. And the idea isn't necessarily, okay, there's 25,000 metabolites that are in the blood. You're detecting 126. That doesn't seem like a lot. Well, the issue is that, again, the redundancy of biology and the fact that disease and changes in your health status do not happen in a one-point manner. You do not get in cancer, decreased levels of citric acid just by itself. You'll get decreased levels of citric acid. You'll get fumaric acid. You'll get, glutamate. You will get the whole sort of Warburg-like effect. And the thing is that disease happens on a pathway scale. So it's irrelevant to ask how many metabolites do you, monitor. What you should ask is how many pathways can you reliably monitor. And with our 126 sentinel metabolites, we can monitor, like I said, 68% of it. So, and that covers the major contributors of the top 6 contributors actually of human disorder, from heart disease down to Alzheimer's.
So if you extrapolated that to like the impact it has, 68% is how much you're measuring, but in terms of like data, like a person actually using it, you're seeing much more than just the 68% because you're categorizing like, it's not about like the super rare disease, even though you can identify those as well. You're categorizing like the biggest diseases and malfunctions out there. So really, you're getting a bigger view than just the 68%, right?
Yeah, I mean,
not in technical terms, but
I know what you mean. I know what you mean.
Yeah.
If you take those pathways that are related to the highest contributors of mortality and you weight them differentially based on their contribution to mortality, sure, you could say that we're still measuring 68% of pathways, but how much of all cause mortality are we measuring? I'm sure it's more than 68%. Yes.
That would be an interesting figure to I don't
calculate
know
it.
if that's super quantifiable, but that would be kind of interesting to figure out.
Yeah, I mean, we can definitely try. I know exactly what you mean, and yeah, that's a great point, Mike. Actually, very insightful. That yeah, we're measuring 68% of pathways, and disease happens at the pathway level, not the metabolite level. And we're measuring 68% of pathways, but some pathways are really idiosyncratic and related to very rare or even super rare diseases. But everything from heart disease through the cancers and Alzheimer's, that accounts for like more than 90% of all human mortality. The others being due to like accident and then some suicide. And so we can catch the ones from disease, and probably that number is well above 68%. And we have this technology, and there's been of course bad actors in the field. I hate to bring up the ghost of Elizabeth Holmes, but Theranos and yeah, and the shadow of Theranos has really cast a pall on this whole industry. Her
What kind
issue.
of test? What kind of test was she proposing?
Great question. So she was doing in vitro diagnostics, and this whole Valid Act is in many ways a response to the based fraud of Elizabeth Holmes. The issue with her was that she didn't have any science. She had no scientific training. If you ever see a company like Theranos or like Therion, and we're trying to like revolutionize the health industry, and the head of that company is not an MD or a PhD, run, run and go the other way. This is simple enough that we can explain to you how it works. You can come into our lab and see your data being generated, but it is not something as such a deep domain knowledge that anybody can do, nor should anyone do. Her biggest thing was that she kind of like modeled herself after like Steve Jobs or Bill Gates, and she dropped out of Stanford, and I have the intelligence to do it. She didn't. This isn't something you can just be smart at. This is something that you need, formalized years of training. It is a practice and a science in many ways. So she had none of that basis. Her next thing was that she just did very small sample sizes on a very few number of people, conjectured that TNF alpha and LDL together created this one diagnostic model, and she sent it off for a patent. Theranos would sent it off for a patent, and then they would patent it, and it was not true. So you would get a lot of false positives, you get a lot of false negatives because the model wasn't validated. What we have are all validated models. We do not employ anything that is not scientifically sound, published in top journals or mined from reliable data and generated ourselves. You do not have that with a company like Theranos. You also had the issue that they were building a literal black box. She wanted to build this little tiny black box called the Edison. And anytime you see a black box, again, run the other way. How do you know what happens in that? And so this thing would break, and it wouldn't even run the test accurately. It was basically a very small clinical chemistry analyzer. And we have one in lab. It's from Beckman Coulter, the AU480. It's a very big machine. It takes up half the room. It's got all these oxygen and water tanks attached to it. It's a really cumbersome thing. She wanted to miniaturize that, and the science or the engineering to support that just was not available. And instead of saying, hey, we're going to use proven methods like a mass spec or a gene sequencing machine, instead of using those, we're going to just sort of build our own thing, completely fraudulent. Investors would come in, she'd show them, okay, take your blood spot on this machine. And then she'd say, okay, let's go to lunch while your results are prepared. And as she would leave, a tech would enter the room, take the blood spot, do the actual bench side, kit-based assay, very old school, put the results back as if the machine had done that. That's not the case. You can come to our lab, you can see how the peaks are generated, how we go from your dry blood spot to a mass spec peak that is, related to, that is the identity of a metabolite, how that's quantified, how we analyze your data using our, Bayesian inference approach. That is all, very important. And Theranos never gave you access to that data. We will give you access to your own data. You can have it. It's yours. It's shared, data agreement ultimately, but you can have it. It's yours. It's transparent. So we believe in a transparent data collection. We believe in transparent results reporting, giving you access to the studies even. If you want to get that sort of level 2 knowledge, that is something that we are working to include in our reports, but we'll definitely have in the app is this, hey, click on this and you can see where are we getting this from? Why are, high levels of phenylalanine associated with mood disorder? Take a look at that. We can give you a very simplified reason and give you how far away you are from the normal range and our confidence and our measurement. We can also give you a very quick blurb as to what that means. And then here's that level 2 data. That is, the real results, the real scientific study or publishing or publish paper that sort of backs up our inference. That's never something that Theranos did. That would have immediately shown their hand and shown Elizabeth Holmes to be a fraud.
Yeah, total frauds. And one thing you did that I was just thinking about, that because we were in Phoenix recently and we were going out to dinner or something, and it's the second night in a row after that meeting, you're like, oh, I made a commitment to help my in-laws move or something. And I was like, that's perfect, because you got to have your home life in balance to do good at business. You can't suck at home and be a rock star. It's just like the type of person you are. Is that spoke to me. Just, it was a small thing, but just in terms of like, we had this like big thing. We felt like we had some momentum going and, opportunity to go out to dinner with some other people and that kind of stuff. And you're like, well, I made a commitment to move furniture. I'm like, that's perfect. That's the way to go.
Yeah, it happens. It happens. You make a commitment to people who you trust. You all work to fulfill your commitments to each other. And, very quickly, you'll see that it's to the benefit of everyone. I try to maintain that relationship with my academic colleagues, with my personal friends and peers who are helping on some levels on, projects in Ethereum. For instance, scaling it to the microbiome. I'm working with, Alex Moore and doing that. And my investors, always maintaining good relationships with my investors, parents, friends. It's all, like you said, it's all in the same ecosystem. And
it is
a great way to judge a person and their overall merits is to see how they operate in their ecosystem. I appreciate that insight. Yeah, I really actually wanted to go out to dinner and hey, let's have fun and talk some more. But yeah.
Well, that's the thing that's admirable about it, is because we're going out with there's something in it for you, and I don't know, the Elizabeth Holmes of the world don't do that.
No.
They say, oh, I have something that is going to advance my career more, or might, so I'm going to choose that over, a commitment I made or family or whatever. So, but getting back to like this, one of my questions is, so like chronic disease shows up in the metabolism far long before the symptoms start to manifest, correct?
Yeah.
So how early, and you might have to take some specific examples, but how early could you detect, say, like cancer or heart disease, or, you can choose one, but before the symptoms actually start to show?
Yeah, absolutely. Great question. And I have a lot of deep experience with this. Early detection is by and large one of the primary goals of creating a new diagnostic. When you look at why new diagnostics are made, there's a few reasons. One is like, hey, it's really expensive, or it's really invasive, or it takes too long to get a result. One major reason is that we're not catching this early enough, whether that's prostate cancer, whether that's breast cancer, valley fever, Alzheimer's. And I have, very relevant experience in this, working in breast cancer and Alzheimer's. I'll give you both examples. When we looked at breast cancer using our metabolomics panel, this was an older panel on a different machines. My first year of my doctoral program, we looked at all stage breast cancer. Breast cancer has a fairly good 5-year survival rate. It's almost 90%. But that number can be as high as 99% if the cancer is caught in stages 1 and 2, very early stage breast cancer. The issue is that current techniques like a mammography, they don't catch stage 1 or 2 tumors. They're necessarily not big enough to be imaged, right? And so we did this test, and we found that, hey, we had like 89% confidence that we could find, breast stage 1 and 2 breast cancer. And so my first ever published study, we looked at 89% predictive accuracy for early stage breast cancer. That was our main goal. We did that. Then we wanted to see if we can increase that number by using less predictors. So initially we had used like 6 metabolites and their age. We wanted to see if we could use less predictors. So we did another study profiling another group of patients, and we found that 3 metabolites using a different panel, a different set of metabolites, 3 other metabolites and their BMI actually was way more predictive, about 96% predictive of early stage breast cancer. We also did a follow-up study. I didn't do this, but it was in our lab with triple negative breast cancer and lipidomics or metabolomics for just lipids, just the lipid species. And we found that not only could we do early stage breast cancer, but we could detect triple negative breast cancer, which is a very aggressive form of breast cancer that is usually not responsive to treatment. And so we found that. That is how we iterate our tests to refine and, again, translate results to hopefully drive meaningful improvements in patient care. The next issue is, with Alzheimer's disease. We had 4 groups. We had normal controls that were completely healthy. We had high pathology controls that had, some pathology of Alzheimer's without symptomology. We had mild cognitive impairment. People who had the pathology had some symptoms, but the symptoms were ultimately consistent with normal aging. And then we had definitively diagnosed patients with Alzheimer's disease. And these are all post-mortem brain tissue samples we collected from these 4 groups. We analyzed the metabolomics. This was a very wide study. So we had 2,080 metabolites in total. And what we were able to come up with was particular biomarkers. We had 4 groups, and we were able to come up with biomarkers for each comparison, right? Each comparison as to which biomarkers should be, swapped out to accurately predict where this person is on this continuum from normal control through high pathology, mild cognitive impairment, and then Alzheimer's. Where are you on this continuum? And we found 4 fatty acids that when swapped out and created different models, we created, 6 different models, I believe, using these 4 metabolites. And with greater than 90% certainty for every group, we were able to say if you have levels of these 3 metabolites below this level, then you are probably normal. Higher than this level, you're high pathology control. These 4 metabolites between normal and mild cognitive impairment, you're normal. We found 1 metabolite, lauric acid, which was a great predictor, 100% accuracy between normal controls and Alzheimer's disease, the 2 ends of our spectrum. And so with careful design, getting data on, okay, what is the pathology? What is the symptomology? We were able to create biomarkers, not just for healthy and Alzheimer's, but also the in-between stages, the prodromal stages or the preclinical stages where the symptoms aren't manifest yet. And that to us is exactly what we want to do in Ethereum. But instead of having, like a study where like we go to a biobank and we collect samples and we buy those samples along with their metadata, patients will report this information, will report this health information. Our subjects or customers will report this health information when they send their samples in. So that when we bank it and then we realize, oh, hey, we have a critical mass of people now with Alzheimer's disease. Let's mine our data and try to find biomarkers and drug targets. We can do that. And we don't need to, do another study or go to another blood bank. Stop doing these sort of separated, discrete studies. And what we need, again, is just one massive undertaking to bring health and medicine into a completely new area where we don't, when you think about what medicine could be, I want you to think about science fiction. Where like people like, if you've seen science fiction, they'll just like be sitting there and they'll say, run a diagnostics on yourself. And it's sort of like a cyborg thing where like the system runs it and it says, oh, you have, high levels of such and such, you need this. We want to build something like that.
Yeah, that's where I was going with this, is what's the future?
That is the future. If you want the future, look at science fiction. That is what we're going toward. And I think, I don't know about the cyborg thing. I don't want to put a chip in anyone, but I do want it to be so easy where you just get on your phone and you say, order this test. It's to your door in a day. you send it back in a day, and the day after, in 3 days, you've got your full results, and it's interactive and it's meaningful, and you can have longitudinal results stored right there and analyzed for you. That doesn't happen when you go to Sonora Quest. They don't bank your data, analyze it, and give you new analyses based on the context of those changes. That never happened at Theranos. I took a Theranos test once, and it was just a piece of paper. It was a piece of paper, and 3 months later, I got a letter from Walgreens telling me that the Theranos test was a fraud, and they refunded me $15 for my copay. That was the life and death of Theranos. It never applied bioinformatics to their results. It never tried to do science. It was always just trying to play catch up. It was faking it till it maybe someday made it. And it was never going to happen. And what we have is this really cool opportunity to bring science fiction to life. It's not going to happen, maybe it will, but it's not going to happen in my lifetime where you have like a cyborg entity that's able to self-diagnose. But we can do very rapid, this telehealth thing, especially after COVID, it's not really going away. And why not have your tests here analyzed for you? And if you want more information, schedule a consult with one of our physicians, in the comfort of your home. And if you need to come in for like a physical examination, which a lot of times that happens, or imaging or something, you can go do that. But that power and that autonomy is put in your hands. We don't believe in, this. I've always said, like, this idea of biohacking. It's a bad way to call it, because it kind of makes it seem like you're doing something you shouldn't be. Like you got your hand in the cookie jar, like you've gotten to something you shouldn't have ever been allowed to get into. And that's fundamentally the wrong premise. This is your biology. This is your data. You deserve to be in charge of it. You deserve to be knowledgeable of it. Wisdom begins with knowledge of the self. That's Aristotle. And in a lot of ways, that philosophy permeates and propels us forward. So going back to the science fiction, we're working on it. And I think very quickly, within our lifetimes, you can get to a point where you put on some virtual reality goggles and you walk through your biology. You interact with your, microbes, you see your metabolites. This could be a total revolution in how we conceive of our health.
Yeah, and I also like that you're not like, hey, I want to put a chip in people. Like you have more correlations to a naturally minded practitioner than I expected, coming from academia. But there is a pretty distinct split between academia and medicine too. Like, I mean, obviously they correlate and they work together, but it seems like it's not as succinct as you would think.
Not at all. It's not as it's not as succinct, and it's not as in sync. The idea is that in academia and research, we develop the techniques, the technologies, the science-based knowledge that is then harnessed by primary care physicians or physicians in general in their clinics. That doesn't always happen. The translation, like I'm saying, that translational throughput is incredibly impeded. There's a great bottleneck, and it goes back to things like inefficiencies in federal funding, or skewed scientific incentives, poor guidance from government guidelines. It's a myriad of reasons for that. But that handoff. We were running this relay race essentially, where we're handing the baton off to the primary care physician. It doesn't happen well enough, and that's not necessarily anyone's fault. Like I said, academia is currently crippled and is actually going down pretty quickly. And that doesn't matter if you're at like an R2, even R3 institution, or if you're in a massive land grant institution like ASU R1, or you're at Harvard, you're in an Ivy League. Academia is being crippled. It's kind of like been shot in both knees, and it's trying to keep up. If it doesn't get fixed pretty soon, I don't think academia will necessarily be the heart of innovation anymore. I think industry is in many ways taking the best people and employing the best processes. And so industry, the pharmaceutical around the scientific industry in general, is going to overtake probably the innovation that's happening in academia. The next issue is that, physicians are busy. And I don't blame them.
Yeah.
Physicians read abstracts. Physicians read the abstract, and if that knowledge isn't, immediately, they're not able to immediately glean that knowledge from the abstract, they have to move on with their time. They have people to care for, right? Like I have test tubes and vials and a whirring machine, and they're all at my convenience. They are there to be used by me, but as the physician, you are the thing being used. You are the person serving other things. And so they don't have the time. What we need is this entity that will say, hey, we will translate these results. We will give you the data used to create these inferences. We will create the test that'll let you recreate this method and take this data from the patient's blood and analyze it and give them a result. And we'll empower you on how to interpret that and how to apply that. That is what we can do with Ethereum. And I think that is what physicians want. They need that translation to be done for them. They do not have the time to do that. There is, a bevy of researchers that work essentially in translational science. They work sort of this bench to bedside, they say. It's a really small group, relatively speaking, and it's not really that efficient, especially metabolomics and systems biology. So, like I said, in many ways, way more ways than one, this mandate of assembling this body of knowledge and delivering actionable results to people, that mandate is best in the hands of a private business entity. Even in ASU, again, without naming names, there are initiatives aimed to do something similar on a narrower scale, on a far less imaginative scale. And they have spent I think $5 million so far. And all they've done is interview community members, getting opinions on what people want. That's $5 million on market research is so that's what I'm saying. This cannot be, I don't want to be a professor writing grants trying to waste taxpayer money on a good idea that was always going to fail. I'd much rather take our chances here and let the markets decide. I think people want this. I think if you explain to them, hey, what you can get here is, if not cheaper, then at the same cost. It's faster, more accurate, far more convenient, more reliable, more accessible. It exists in perpetuity on your phone. All of these things I think are so attractive to your average consumer, from the guy who is, very health conscious, health nut, to the guy who hasn't been to a doctor in 5 years because he hates it. On the whole scale, not whole continuum, I think what we're offering is very attractive, and that is what people want. They don't want to be interviewed about, what's your health like and what health challenges does your community face. That's up to you. My goal is to look at what you give you an interpretation and put some power and autonomy in your hands.
Yeah, it's just saying, hey, here's what's happening. I wish we could talk for 3 hours, but I got one last question for you. We'll probably do some more podcasts or webinars or something and just have continued conversation, because you just have such a wealth of knowledge. And it's fun for me to learn all this stuff, because you have just a very different mind than a lot of people that I work with. And it's really cool just to be introduced to somebody as passionate as you are, that, exists in academia and all that good stuff. So let's pretend, though, that this product that we're imagining, Ethereum, is operational. So that might be 5 years down the road, it's successful, right? And then another 10 years past that, it's now standard of care. So maybe 10, 15 years out. What happens? What happens to medicine? What does that look like?
Yeah, so it approaches, if we become standard of care, and the government has a centralized patient system where you can have your data there, and then it goes between clinics, or should you try to transfer it? We want to be that system. And if it becomes in 10 years of standard of care, we will necessarily become that system where, if you look at, health databases, the government has been doing this NHANES survey since, I don't know when, the 80s, perhaps. When you look at what's the greatest wealth of current anthropometric and physical activity data, it's not the NHANES, which has been going on for 30 years. It's Apple. Apple and Amazon both sit on vast wealth of human physical activity, nutrition, movement data than NHANES and the government system ever could hope to achieve. And it's objectively measured, not self-reported. And so we can do that. The same thing on the molecular level, right? There's no way your Apple Watch is going to give you 126 metabolites in our lifetime. There's no way your Fitbit is going to analyze your genome in our lifetime. But we can do that on a molecular scale, and we can totally make what the government has been trying to do obsolete. And I think if you said in 10 years become the standard of care, we will do that. What happens then is a rapid advancement in clinical care because of the data we have. Insights come from data. Like it does. Data itself doesn't equal insight. You have to do things to that data, but you need a lot of it, and as time goes on, things will change. Our data analysis methods will improve. So we may need less data to get the same result. Or our understanding of the biology in some ways might shift such that the data we collected are maybe less or more relevant. All things being relatively equal, though, what we should see is hundreds, if not thousands, of discoveries coming out of this data set. Validations of previous findings, and as well as identifying, really massive data-driven hypotheses for future experimental work. This is going to be sort of like the Rosetta Stone of human biology. If this is done correctly, this is the Rosetta Stone.
Mm-hmm. Yeah, I mean, you'll have better drugs, getting rid of stuff that doesn't work, finding stuff that does work, early detection, all those things. How long do you think people can live?
Aging research is a big part of my work, actually.
Paniz is going to figure it out.
I can say so much about aging, and the overall disease process of aging. But yeah, we are looking at improving, lifespan and quality of life. We are looking to empower people with their health data. It's no longer this, Peter Thiel says, what's your big secret? Our big secret is that, health and science is what we're using today in current medicine. It's obsolete. It's like we're using, techniques from 50, 60 years ago still to diagnose. And when we know that, these sorts of tests inform 70% of all of our clinical decision-making, and they only account for about 6% of all our spending, and that we're using methods from still necessarily, the time of when antibiotics were discovered, then you realize that we're doing medicine in the Stone Age. And we have this sort of technology in the ivory towers of academia. And it just takes, one Prometheus, one Titan, to steal that fire and bring it down to people and say, hey, we're going to turn our instruments customer facing, and we're going to drive healthcare rather than some more publications, 3 more publications for me this year. That is an irrelevant marker. We need to move the goalposts, very much so, in science. And we've lost the forest for the trees, and we need to focus on people again.
Yeah, I love it. So where can people find more information if they want to?
Okay, so right now we've sold out of our first phase 120 beta testing kits. But if you'd like to go check out more on us, our team, our science, mission, vision, values, please go check out www.therio.me. That's therio.me to check out our website on Theriome. You can also contact me via Jasbi, JASBI, at therio.me, or connect with us on Twitter at Real Theriome or at Dr. Jasbi. And I look forward to getting some feedback from your audience, Micah.
Thanks, Paniz.
I appreciate it, Micah. Thank you so much, and we'll be in touch very soon, I hope.